Imagine trying to, like, reverse engineer the entire operating system of the human body, right? And then you find out the core kernel is written with an alphabet of literally just 4 characters.
Yeah, it's pretty mind-blowing when you think about it that way.
It really is. Okay, let's unpack this. Today we're doing a deep dive into the 1953 discovery of DNA's structure. We've got some fascinating source material, and our mission is to figure out if our genetic code is, our absolute unchangeable destiny, or if it's something we can actually rewrite.
Right, and that whole tension between destiny and editability, it really burst into the public eye back in 1953, specifically at the Eagle Pub in Cambridge.
Oh, right, the famous pub boast.
Exactly. James Watson and Francis Crick walked in and just loudly announced to the room that they had found the secret of life.
I mean, that's bold, but they
Okay.
actually had the physical model to back it up.
They did, yeah.
Because, anyone following biology knows those basic 4 chemical letters, right? Adenine, thymine, guanine, and cytosine. And that A always pairs with T and G with C.
Right, but what's fascinating here is that the breakthrough wasn't just, listing those chemical ingredients. It was realizing how the sheer geometry of that exact complementary pairing perfectly solved this massive biological mystery.
The mystery of how cells unzip and copy information without just descending into total chaos.
Exactly. It wasn't just a chemical quirk, it acted like a mechanical zipper. When the molecule unzips, those strict geometric constraints force each half to rebuild its exact missing complement.
Oh, I see. So it's kind of like a self-replicating spiral staircase.
That's a really great way to put it.
But if it's just a 4-letter alphabet, why did it take a genius breakthrough to realize how it folded together?
Well, because figuring out that complex shape wasn't just a two-man job. It required seeing the invisible, basically.
Which is where Rosalind Franklin comes in, right, with
Yeah.
photograph 51.
Yes. Her X-ray diffraction image was the vital clue for the double helix.
Because it wasn't a regular photograph, it was like an X-ray scattering off the crystallized DNA, leaving this distinct X shape of black dots, which mathematically proves it's a helix.
Right, and that image gave the exact mathematical parameters Watson and Crick needed.
Yeah.
She died before the 1962 Nobel Prize went to Watson, Crick, and Maurice Wilkins.
So what does this all mean? I mean, for how scientific credit is handed out and how data can just be overlooked?
It's a tough historical reality. But if we connect this to the bigger picture, this single discovery fundamentally shifted biology from being a mystery to a concrete information system.
Which ultimately birthed the 2003 Human Genome Project.
Exactly. And modern forensics and basically all of modern biotechnology.
Because we mapped that structure, we went from merely reading the code to wondering if we can actually rewrite it.
Yeah, but DNA interacts heavily with your environment and your lifestyle, It's really a risk indicator, not a definitive death sentence.
Here's where it gets really interesting for you listening. If the genome isn't just a simple text file we can casually, like backspace and edit, how do we safely fix genetic diseases without breaking the entire biological system?
That is the million-dollar question. We can predict risks and explore gene therapy, but intervening requires extreme caution because of things like pleiotropy, where one single gene affects multiple totally unrelated traits.
Oh, so intervening is incredibly risky.
Very risky. Which ties directly to you understanding that those commercial DNA tests just offer statistical probabilities, not an unavoidable fate.
So we've taken this profound leap from a 1953 pub declaration to today's frontiers of gene therapy. We've gone from marveling at the ultimate instruction manual to holding the pen.
We really have.
But before we start aggressively rewriting our code, it leaves you with a pretty massive thought to mull over. If humanity eventually perfects the ability to rewrite our genetic code, to erase all hereditary diseases.
Which sounds like an absolute dream.
Right. But could we accidentally eliminate the random genetic mutations that actually drive human evolution and adaptation in the first place? Like, if we erase all the errors, do we stop writing new chapters?
That is, yeah, that's a heavy question.
Definitely something to think about.